So chapter 15, we focus on the third line of defense. If you recall, in the previous chapter, chapter 14, we looked mainly at the first two lines. We talked about the first line of defense, remember, was those barriers and the chemicals that help to protect the portals of entry. The second line of defense, remember, we talked about the cells themselves, like white blood cells, leukocytes, and the chemicals that can be produced from them. And so in chapter 15, we focus on the third line of defense, which is that adaptive or acquired immunity. And so remember, the main feature of this line of defense is that it only happens through prior exposure. So this type of defense mechanism, it is acquired over time. So it's responsible over time for producing long-term protection that you develop through exposure. And this exposure could be through infections or through vaccinations. This type of immunity also is centered around what we call B and T lymphocytes and the cells, or you could also call B and T cells. And so if we look at just a couple quick vocabulary words that are associated with these, and I will go ahead and preface to say this, in Anatomy 2, you go into a lot more detail into this third line of defense, and your textbook does as well. So starting on page 468, it goes through all the parts of the specific immunities. In micro, we don't have the time to go into all that great detail. So this may be one of the chapters where you have to spend some time reading through some of the content we don't cover in this PowerPoint if there is some confusion that's there. So we're just going to kind of skim the surface on a couple of the most important things. So one vocabulary word that's really important we talk a lot about with specific immunities is immunocompetence. Immunocompetence is your ability to be competent enough that you can interact with foreign substances. And so this actually starts there in fetal development, and as you continue to grow and you get towards puberty, even late puberty, that's where this becomes completed. So immunocompetent is just the ability for your cells to recognize self from foreign. And so this is a process that you have to kind of work through. You're not automatically immunocompetent. It's something that happens again through experience, through exposure over time. And so another vocabulary word that we mention here is something called an antigen. So what an antigen is it's a molecule that stimulates an immune response. Okay, so typically consisting of proteins, there's some polysaccharides that are there, maybe some other compounds, but an antigen is any type of molecule that stimulates a response. So it mentions at the bottom of the screen, it even could include environmental chemicals depending on the fact that, again, they stimulate an immune response. Now, a couple quick vocabulary words that we want to mention in response to specific immunities. Two characteristics of specific immunity. Number one, the specific part. So specificity. What happens through exposure is your body produces antibodies. These antibodies function only against the specific antigen to which they were produced to respond to. For example, chickenpox virus antibodies only respond to chickenpox virus. They will not respond to the measles virus. So even though they're both viral, again, antibodies are antigen specific. So that's what specificity means there. The other word that they use to characterize specific immunity is memory. See, what happens is once you produce an antibody, lymphocytes are programmed to recall their first encounter with an antigen. So when they come in contact, when your antibodies, when your cells, when they come in contact with the antigen, it automatically triggers a memory of when they met that antigen before, of the prior experience they have with that antigen. And what the memory allows you to do is to recognize it faster and respond quicker so that you can keep an infection from occurring. So two really important things that we use to characterize specific immunity with. So coming back to an antigen, we want to look at some specific characteristics about an antigen and kind of go from there. So remember, antigen is any substance that elicits an immune response. So it's any type of material, any type of molecule that you're going to respond to. So antigenicity is just a property of behaving as an antigen. So it's a property of causing a response. Now, one of the things they mention in chapter 15 is that the terms antigen and immunogen a lot of times are used kind of in exchange of the other. but they're technically not the same thing. So an immunogen is a type of antigen that actually induces an immune response. So what they tell you is basically some antigens, they don't fit all the conditions to stimulate an immune response. So what conditions are we talking about? Well, size, shape, accessibility, and foreignness. And so if an antigen doesn't meet all those conditions, then it may not actually trigger an immune response. If it does trigger an immune response, we call it an immunogen. If it doesn't, we just simply call it an antigen. I'll give you an example. So there's a certain type of bacteria, it's called Haemophilus influenzae. Obviously, it's one of the ones that can lead to certain type of the flu. And so they mentioned that there are small polysaccharides that are foreign. They're antigenic, so they should trigger an immune response, but they're too small to actually do it. So their size kept them from being an immunogen, so one that actually triggered an immune response. So they talk a little bit about size and the fact that they need to have typically a molecular weight of like 10,000 to be actually an immunogen to cause an immune response. So if you look at the picture, just again, this is kind of an Anatomy 2 lesson, but so this is what a typical antibody looks like. And so they mention that what antibodies do is they typically recognize and respond only a portion of the antigen. And so this portion of the antigen is called the epitope. And so again, here's your antibody. This area here and over here are known as the antigen binding site. But again, you'll notice it's not very large. It is only the size for that fragment. So the certain epitope would bind in here, and if it binds and fits in, then that antibody knows and recognizes that this is a part of that antigen that it needs to carry out an immune response for. Okay, so this is where that antigen binds to, and again, it's not the entire antigen that binds, it's just a part of it. It's called the epitope. And if it binds in and it fits like a key in a lock, then that's going to then trigger an immune response. They also mention haptens. So haptens, a term that we talk about in Anatomy 2 as well. They're very small, and by themselves, they're too small to elicit an immune response. So what happens is sometimes it'll get bound to a carrier molecule, and the carrier molecule is what actually allows it to trigger the immune response. So just a vocabulary word that's listed there. They talk about some different categories of antigens like alloantigens and super antigens. I did want to mention the bottom two that are listed here because we're going to come back to these in Chapter 6. But allergens. Allergens are actually antigens that evoke an allergic response. So this could be, and when we think about all the different materials that can trigger an allergic response, so these could be chemicals found in food, right? This could be stuff found in the air that we're breathing in. This could be venom from a sting, right, from a bite that we get. And so a large range of allergens, which are actually antigens because they elicit an immune response. They also mention autoantigens. So autoantigens are molecules in your body for which tolerance is inadequate, meaning they actually attack yourself. And so we talk about autoimmune diseases in Chapter 16. We'll come back to those guys.